hello everyone, and a very warm welcome to our second episode of the Live from the Lab Stories of Animal Ancestors series. Today, we will get an insight into the microscopic and molecular world of scientists that are studying the evolution of cell types. My name is Siri Kellner, and I am doing my PhD project here at the Museum für Naturkunde. With that, I would like to say a thank you to the museum for giving us this opportunity for hosting this event, but also a thank you to the experimental field who is giving me this room here right now, and also for the virtual room of the Zoom account. And with that, I would like now to pass you over to Milena, who organized this event with me.
Hello, everyone. I'm Milena. I'm doing my PhD at the University of Exeter, and I'm going to be the moderator for tonight. Me and Siri and all our today's speakers are doing our research within EvoCell, which is a Marie Curie network gathered around the theme of animal evolution from a cell type perspective. And you had the chance to see what that means in the trailer that I showed earlier. So we prepared it to relay the message that all the different animals that we will talk about in this series, which are mostly obscure marine creatures, have very similar cell types and that they all come from the same ancestor. And today we have 4 speakers who are all fellows in the EvoCell network, as I said, and we will start with Luca and Francesca, who will lead us through how animals evolved from single-cell organisms and then how in their development they make all the different cell types. After that, we have Petra, who will talk about reproductive cells, and our last speaker will be Alba, who will talk about how regeneration works in animals that can make their whole legs grow back. And our speakers today, they have prepared their talks as a team, so I'll leave it to them to introduce each other. Our first speaker is Luca from EMBL, and Luca, you can take it away from here.
Okay, then I will start. Hello, everyone. Good evening. And yeah, I am Luca, live today from Germany. And yeah, I was born and lived most of my life in these beautiful places in Italy. And then 2 years ago, I moved to Germany to study the evolution of cell types. And here at EMBL in the middle of the forest in the lab of Detlef Arendt. And I will try to explain you in the next 5 minutes what does it mean to study evolution of cell types. So this is our planet, this immense spaceship, and we share this place with astonishing variety of animals with all sort of exotic shapes and colors and dimensions. And yeah, and they look very different, but despite all their difference, actually they have a lot of things in common. One of these things are their structural microscopic components, cells, which is the smallest unit of life on this planet. And animals have a vast variety of cells, each one specialized, highly specialized for a specific function. Some of these cells can be very similar between animals, and the information to make them, it's all encoded inside their DNA. So why do animals have those things in common, all these things in common? Well, the reason is that they all have a common ancestor at some point in the past. So for example, according to this tree, if we want to find the common ancestor between like an earthworm or an octopus, we will have to go here. But if we want, for example, to find their common ancestor with a fly, we will have to go far back, and even farther back if we want to find the common ancestor with a vertebrate like a whale, a bird, us humans, we will have to go here. And we can go way, way, way far back until at a certain point, probably we will find a unicellular organism from which all these animals descend. Now, in the next slide, I would like just to show you sort of a speculation. It's just to very easy schematic, just to make clear how the concept of how new cell types might evolve. So let's imagine we can go back in time, like to some 600 millions of years ago, using our favorite time machine. And we'll probably find in the ocean this unicellular organism. We don't know much about its biology, and this is why it's out of focus. And so this organism has to do a lot of tasks in a single multifunctional unicellular compartment, which is quite impressive and complex to achieve. But we can imagine that at a certain point, some of these organisms, maybe after cellular duplication, started cooperating and stick together. And then some cells started migrating inside. And maybe at the next step, we can already start, cells maybe already started dividing their tasks. So starting specialized for a certain function. And we can maybe identify some cells that became specialized to control the water current, and the one that are in the inside surface, they started getting specialized to absorb nutrients, to metabolize nutrients. And then the one that instead are exposed to the surface started making sort of an epithelium, and then cilia maybe became specialized in order to perceive the external environment, like changes in pressure, light, or chemicals to detect food. And then some of these animals might have become bigger, and then we need like some coordination between all the body parts. So maybe some primordial neurons and contractile cell might have appeared. And then maybe we need some more coordinated movements. So maybe some more specialized muscles started to appear, and then some more neurons, et cetera, until we see all the biodiversity of the animals today. And the animals today can have hundreds of different cell types. And some of these cell types can be very specific to these animals. For example, some of you might have experienced the piercing cnidocytes of jellyfish, of the tentacles of jellyfish, for example. Now, as I mentioned before, all the information to make all these different cell types is all encoded inside a molecule called DNA. Now, the thing is that all the cells in your body, they have exactly the same copy of DNA. So how do we make hundreds of cell types if each cell has the same information? So cells use some complex regulatory mechanism in order to access just the information that they need. And yeah, they can be of various types. Some of them can be like the state of the DNA that can be tightly packed together with proteins, so that is not accessible. Some other regions of DNA can have more of an open structure, so it can be bounded by some specific proteins that we call transcription factors, and they have a specific affinity just to some sequences on the DNA. So the combination of all these regulatory mechanisms can, in some can give rise either to a muscle type, and then if we have another combination, we can get like a gut cell, and with another combination, we can have a neuron, for example. And so we study all this regulatory mechanism, and we try to compare them across all the species in order to understand what, for example, what do equivalent cell types have in common in all these species, if they had a common origin in the past. And so this is how we try to understand the evolution of cell types. And now I will just show you, let me switch to the other camera. Okay, hello. And I will show you some tools that we use to study these things. So here are, for example, the animals that I study. These are marine anneli. They're small worms, you can see, and usually I study their kids. So you can see here on the slide that black dot. They are very small, they are larvae, and they are 10 times smaller than that dot that you can see there. So, and this is instead This is Mr. Microscope, confocal microscope. You see there is a slide, and here is the screen. For example, here I stained this larvae with fluorescent molecule that can detect just some molecules that are activated only in muscles. So here you can see now it's scanning, and in blue you can see here is the head, in blue are all the cells, and these are all the muscles. You see how specific is this staining, and then we can also zoom in, for example, in the leg here. No, it doesn't. Is it working? No, it's not working right now. Here, yeah, you see, and we can see all the cells with the staining. And I can use more or less the same fluorescent molecule to stain also the muscles in your body. Okay, so now we'll go back to the other camera. Cool. And I will introduce you to the next speaker. Give me just a second. Okay, cool. So we will talk about also these other topics today, and the next topic is the research topic of Francesca, and is that process for which animals at each generation have to rebuild the entire body, starting from a single cell. And we all have been that single cell at the very beginning of our life. So Francesca, are you ready to go?
Yes, so hi everyone.
Cool.
I'm Francesca. I'm also Italian, but now I'm in Norway, in Bergen, in a lab that works on comparative developmental biology. And I look at gene expression in single cells. And if you have no idea what this is about, we will get there. So, as Luca just said, all our lives start from a single cell, the zygote, which is just an egg that has encountered and been fertilized by a sperm. And this single cell starts dividing into 2 cells, and then 4, and then 8, 16, and so on, until we get to a very high number of cells that start organizing themselves in groups and moving around the embryo to form different structures, like we can see in this video of a frog embryo. In this process, which is called development, we get from a single cell to a fully formed animal that has different cell types. So some Luca has mentioned, that you probably know them. So for example, neurons, which receive inputs and deliver outputs to the outside, but also to our body. And then we have muscle cells that we need to move, and gut cells to digest and absorb nutrients, and also adipocytes, which are the fat cells that store fat for the dark times. And all of these cells come from a single one. How can this be? As Luca already mentioned, all the cells in an organism have the same DNA, but they can only access and use different bits of this DNA, which are the genes. And during development, when there are a lot of cells in an embryo, at some point, some cells start to turn on some genes and other cells turn on other genes. And the list of genes a cell type, a cell turns on, is called its gene expression. And the gene expression of a cell determines its function and its type, so its cell type. But different animals do this at different times during development. And so they have different cell types, but they also, these cell types also appear at different times during development. And if we want to know what the ancestor of all animals looked like, which cell types it had, and how these cell types would arise during development, what we need to do is look at the existing animals and compare their cell types in their development. So Luca already mentioned this, and Alba will talk about this later. But if you remember, I said that in my lab, we do comparative developmental biology. So this is it, we compare development. And what am I looking at? What am I working with? So this weird animal over here, which is called Convolutriloba macropiga, which is a very long name, I agree. It took me 1 month to learn it. So let's give it a nickname and call it Connie for this talk. So Connie here is a tropical animal, and we keep them in aquarium that you can see behind this door. So I cannot bring you in because the connection doesn't work, but I can show you a picture of this aquarium here. And they think it's big, but they're actually super small, like half of my pinky fingernail. And an interesting fact about Connie is that it can reproduce in 2 ways. So one is sexual, like us and the frog embryo I showed you before. So from one cell, we get to a fully formed animal because the cell divides and starts expressing genes, and we get different cell types. But they can also reproduce asexually, which is what this guy over here is doing. So this Connie over here is producing a new individual, and this bud here will grow, and for all the cell types of Connie, and at the end, when it's grown, enough grown and looks like a fully formed animal, it will detach, and we will have 2 Connies, identical but distinct. This is interesting, right? But I said I am looking at cell types. So what cell types are there in this animal? Let's start with the green cells that we can see all around. Well, surprise, they are not Connie cells. They are actually algae that are in symbiosis with this animal, which means that the algae and Connie cannot live apart. They need each other to survive, like us and the bacteria in our guts. Then we can see red cells over here, which are gland cells that produce and release substances. And well, Connie moves, so it must have muscle cells and neurons, and that's the case. And then it has skin cells, which are covered in tiny hair. And these are only some of the cells that Connie has, and they have all been discovered by looking at them under the microscope because they have different shapes and colors. But I told you I'm looking at gene expression. So what genes, what bits of DNA are turned on in different cells. And this can give us different information because some cells can look the same, but they can have different functions and different identities. And to see this, we really need to look at gene expression. This is what I do. So this is all for me, and you can ask questions later. And now I'll leave you back to Luca for the next.
Okay. Thank you very much, Francesca. Super nice. So now, yeah, the next topic is here. So yeah, now we're gonna talk about a very special cell types, very, very important because they are the ones responsible of carrying all this information accumulated in millions of years to make animals. Petra loves these cells. So Petra, do you want to talk us about them a bit more?
Yes, thank you, Luca. Good evening, I'm Petra and I come from Slovakia, but I'm currently based in Bergen, Norway. I'm a PhD student at the University of Bergen and I work in the same lab as Francesca does. So yes, I also compare development in different animals, but I'm specifically interested in one cell type that Luca already outlined. And this is the one that's responsible and it enables for development to start and then repeat it again, germ cells, which is what I want to talk about today. But first, let me get your opinion on how I approach this topic. What do you imagine when I say germ cells are real gems? Is it a good metaphor? Do you see what it's based on or not really? Why don't you let me know in the chat? I'm really curious if I should just keep using this metaphor and if it makes sense or if it doesn't. So help me out here and drop a message in the chat saying yay or nay, yes, no. Also the ones watching from YouTube, you can comment in the comment section. If you just are willing to help a sister out here, because I really don't know if this metaphor makes sense anymore. Yay. Okay, well, some of you are of the same opinion as I am, which is good. Okay, a lot of you are. Okay, but I think that most of the people can't really appreciate how special these cell types are, or can't really imagine what the germ cell is. So I'm going to spend the next few minutes explaining what they are. And why would I even compare germ cells to precious stones, right? Why are they so special? Well, because they simply are. You and I both wouldn't be here if it weren't for them, right? And you may be so smart and say, well, we also wouldn't be here if it weren't for the bunch of nerve cells in our brains reminding us of these events at night. And you're absolutely right. But as sexually reproducing organisms, all of us needed that sperm and egg to create an embryo at the beginning of our stories. And only then could we have more specialized cell types in our bodies. So we are very much obliged to gems, right? You start to see it now. Like I said, only then could we have more specialized cell types and bunch of neurons, muscles, and all different cells. So this means they are the only cells in your body that do not care about you. Yes, friends, I'm sorry to break it to you. They do not care about you. All they care about is making it out of your body to start a new life, germinate, much like seeds do when they form a plant. But this also means that they are the only cells carrying genetic material to the next generation. And hence, they are the cells driving the evolution forward. And you can imagine that this was a big deal for early multicellular organisms, and because they had to make sure that only these special cells would be the one passing the message on, and not the body cells. So what we often see in the animal kingdom is the animals make a clear cut between body cells and gems to make sure that they don't mix up and the information doesn't get crooked, right? And then the gems are stored in a special reproductive organs where they are happily rested until their time arrives, much like family jewels in a treasure box. And I study this phenomenon in the animal that is very much similar to Francesca's. It's a tiny marine worm called Meana, and it lives inside of sea cucumber. Well, because of its life cycle is really complicated, we have to go out every season and collect it in the wild. So we can't really keep it in the lab. So what we still don't know about Meana, this hermaphroditic worm, is where its gems come from and when do they arise. We know that it's hermaphroditic, which means that it has male and female gems, but when do they appear? How do they appear? All these questions I'm trying to figure out in my research. And also, how is it possible that some animals can make it in evolutionary terms after their gems are lost? They can regenerate the germline, or they can reproduce asexually, much like the animal that Francesca just introduced. Lucky for you, we also went out this year to collect Meana, and so now I'm able to show you its juveniles, its offsprings that are just 2 months old. And while I set up the camera and the microscope behind me, why don't you let me know in the chat if I managed to convince you that the germ cells are real gems. So I really hope that now you begin to appreciate them more. And there it is. And introduce you to a 2-month-old baby of my marine worm that is much tinier than Francesca's or Luca's. So you can see there's a huge variety of what we all call
a marine worm. And let me know if you can see it moving. There are a bunch of cells around that move. And you can see that in the core here, there's this ball that's slightly lighter than the rest. And that's a juvenile that will hatch out of this membrane soon. So thank you for your attention. And back to you, Luca.
Thank you very much, Petra. Okay, so now we will go to our last speaker, Alba, with my favorite topic, regeneration. Yeah, so unfortunately, as humans, we have a limited capability of regeneration, but imagine from a moment that you can regrow your lost arm. There are some animals that can do that actually, and Alba will talk more about this now. Are you ready, Alba?
Yes, thanks, Luca.
Good.
Hello, everyone. My name is Alba Almazán. I come from Spain, and I'm doing my PhD in Lyon, in France. And today I'm going to talk to you about animal regeneration. So regeneration is the ability to regrow body parts that were damaged or cut off by accident. Maybe some of you remember when you were kids have seen a lizard without a tail or with one that looks new. And for me, it was the first time that I realized that some animals can regenerate. But of course, lizards are not the only ones. Salamanders, spiders, crabs, and octopuses can also regenerate their extremities or limbs. And some animals can regenerate their internal organs. For example, sea cucumbers can regenerate their gut and zebrafishes their heart. That is already quite impressive, but there are some animals that are able to regenerate the whole body from a tiny fragment. This is the case, for example, of starfishes regrowing out of an arm, Hydra, not the mythological creature, but this aquatic animal, and flatworms. The latter ones, you can cut them in 100 pieces and you will end up with 100 flatworms. And interestingly, they do the same for asexual reproduction. So we all can imagine which are the advantages of having such an ability. For example, coming back to lizards, when they are attacked by a predator, they can just drop their tail, which will continue to wiggle, and the predator will be distracted so the lizard can get away. But how does regeneration happen when we look into the bodies of these animals that are regenerating? Well, some animals have a population of cells that are especially set aside for this purpose. They are known as stem cells. And when the animal loses a part of their body, these cells will be activated, start to multiply, and turn into the various cells that make up the regenerated structure. But what if the animal does not have this population of stem cells? Well, in these cases, normal cells that happen to lie close to the wound will lose their identity and will become something similar to stem cells, because normal cells cannot multiply, but these ones can. So they gain this ability and reveal all the missing tissues. So we have seen that while some animals can regenerate, others not. Among those who can, they don't all regenerate to the same extent or don't do it in the same way. And this variety or diversity can make us think that there is nothing in common and will imply that the ability to regenerate have appeared multiple times independently during the evolution of animals. Is that true? Well, we don't know yet. And this is the big mystery about regeneration. So how we can know this? We can use comparative methods, as Luca explained at the beginning. And this is a very powerful approach that we have to understand evolution. So in practice, what we can do is to study this process in detail in as many animals as we can, and then make comparisons. And if we find similarities, maybe that could imply that this ability was already present in the common ancestor of this animal, so it was already able to regenerate. In my case, I'm studying a very tiny shrimp, which name is Pariale hawaiiensis. And this animal, of course, can regenerate. They regenerate all the appendages. This means they can regenerate the antennae, the claws, and the legs or limbs. Their bodies are covered by an exoskeleton, which is a sort of armor, and it will be shed and replaced as the animal grows in a process known as molting, which, like you can see here, a picture of this remaining armor to the animal mold, which is nice and creepy at the same time. In Pariale, when Pariale loses a part of the bodies or a part of the legs, the new one will be regenerated within this exoskeleton. And it will be only visible after the animal molt. We have tools in the lab to visualize this and follow this process. For example, I'm going to show you a very cool video made by one of my colleagues, and you will see that cells are labeled with fluorescent. These allow us to track the whole process. So regeneration in Pariale takes around 1 week. And what you are seeing here, what is happening is that the tissue has to retract a bit to create enough space for those cells that are multiplying and shaping the parts that are missing. So by the end of the movie, you will see all these missing parts will be here encapsulated in this exoskeleton that they will be free by the mold. So we can study this process in this way, but I'm taking another approach for my project. This is based in a very new technology and the concept that Francesca told us, we can distinguish cell types by the genes that are activated in those cells or by the gene expression. So with this powerful new technique, we can gather this information for thousands of cells individually. And then we take this big amount of data and I usually use computational methods to analyze it. In this way, I can take this multidimensional data and collapse it into dimensions to show you a graphic like this. Here, each dot represents one cell. And all of these are the cells that we can find in Pariale limbs. So really, cells that are cells that belong to the same type or that are very similar will be placed very close, and it will share also the same color code. And in this way, we can identify some cell types. This is, for example, the ones that the actual we find in Pariale. But at the end, this is just a snapshot. And what I want is to find the responsible for generating the new links. So to do so, I'm going to take this snapshot during different times along regeneration, and then pull this information and see the relationship between the cells at the beginning and the cells at the end. And this way, identify the ones that are responsible for creating the new ones. Not only that, because this is a very powerful technique that gives you a lot of information, the idea is also to compare these cells with other animals that are also able to regenerate and see whether they have something in common or not. And this is everything from my side, so I give it back to Milena.
Thank you, Alba, and thank you, everyone. That was really, really nice and really diverse topics. So we have now 20 minutes minimum for questions from everyone in the audience. So please write your questions in the chat, either on Zoom or on YouTube. And while we wait for the questions to come, I have some questions of my own. And so Alba, I'd go with your talk first. And what I want to ask you is, using these techniques, do you find that Pariale regenerates all of the cell types that it has in the limbs, because it has to be multiple cell types that build a leg? And what do you know about how this works in other animals? Do they regenerate all their cell types?
Thanks, Milena. So, okay, what we know about other animals, because when we see regenerated structures, morphologically they look identical, but with this technique we can really ask whether they are the same cell types that were present at the beginning. And it is known, for example, in lizards, when they regenerate their tails, they don't regenerate the bone. So instead of bone, it has a cartilage. So we asked this question with parialy using this technique. And I used the same as I show you for legs that were regenerated. And what I could see is that all the information that I can get at the beginning, and I say information because we are still trying to identify all the different cell types that are present, but this information is already present when we look after regeneration. So it looks like they faithfully regenerate everything that was already present.
Really cool, thank you.
Okay, I will just go on with the next question. I would like to ask Francesca. So Francesca, you just recently started your project, and I know that you previously worked in a different lab with the different organisms, a very famous model organism, the Drosophila fly. So it's a very well-studied organism. And in your recent research, how you're studying an animal that is less known, you call it Connie. Does this make your research more difficult because there's not much known, or how you feel about it?
Hi, so yes, in a way it's more difficult, but as you said, we have these organisms that we know a lot about. We call them the model organisms, like the fruit fly that I worked with, but also mouse or chicken, and other animals we don't know anything about. And in a way, it's very hard to work with them because nobody has worked with them. So sometimes we can't even get them to live in the lab, like Petra's organisms. But then we can discover much more in a way because we don't know anything about them. So everything we find is new. Yes, it's harder, but I also think it's exciting. But the model organism can help us learn about other things. So we can start from what we already know and then go on and ask questions like modeling, for example, diseases or other things. While with these animals, you can really understand what the biodiversity out there is and how it evolved.
Cool, I agree with that completely. Non-conventional model organisms are the best. We have some questions from YouTube now, and the first one is for Alba, and the YouTube people are asking you, why do some organisms have the ability to regenerate while others don't?
Okay, this is a difficult question, and I don't have a definite answer, but I can speculate, of course. So to me, what I think is that there are multiple theories about the origin of this trait. And for me, I think that maybe it was already present at the beginning of in the early ancestors. And if you are curious, I can talk about this later. But at the end, this ability, our regenerative structure has a huge cost of energy. So maybe it's not always a good strategy, meaning that, for example, us that we are really big, to regenerate that leg will take a lot of energy from us, but also it will take a lot of time. And now we are in the comfort of living rooms, but maybe when we were out there in the jungle, it was even easier just to create, just to close the wound and not spend that amount of energy on that. And for some animals could be also the same.
Yeah, that makes sense. I'm imagining like an elephant trying to regenerate a leg, and that would have to take years, all those cells. So another question / request from YouTube is for Luca. They want to see more pictures from the microscope, and I would add to that maybe you can explain a little more in depth what exactly you're doing on the microscope, what technique you're using there.
Yeah, of course. Let me change camera. Okay, cool. Nice. Okay, yeah, so yeah, we can go here again. I will put, okay, now it's scanning. And maybe I can zoom in some interesting areas. So let's see, like, let's look here. Yeah, there you go. And here you can see these are the nuclei of the cells. I don't know if you can see it in details, but you can even appreciate those regions that I was telling to you before of DNA that is compacted. So you can appreciate that here. And here we are actually at this is the mouth of this larva, and you can see here all the muscle structure, and you see how intense is this staining, because they are producing a lot of this transcript. These are messenger RNA. I think we are hearing more about with the new vaccines that are coming out. So there is the information that is transported outside of the nuclei from the DNA. It's going inside inside the main compartment of the cell. So in fact, here you can see that the nuclei are poorly stained. Yeah, and we can also zoom out again. Let's see. Yeah, here you can see all the muscle structure around the head, around the mouth. Cool. And here there are all the cells in the brain, and I can also go through different sections of it. So you see that the structure is changing. And here I'm going through the animal. Cool. Yeah, I can also show you maybe another picture, another cool picture. For example, here you can see this is another gene instead. You can see that the staining is completely different. The pattern is completely different. And here you can even see like single cells expressing. If you see here, these are all the nuclei, and you can appreciate all the single cell expressing, so specific cell types expressing this particular gene. Yeah, I hope that was enough.
Good stuff. Thank you. So this is possibly the best question ever in the Zoom chat for Alba. I really love it. Thank you, Sarah, for this question. So it says, you were talking about one living thing. I think it was Hydra. She says that she forgot what it was, that you could cut into 100 pieces and get 100 organisms out of it. Is it impossible to kill it by cutting? And what is the minimum amount of material needed to survive for it?
So yeah, I also love the question. It was not Hydra, it was flatworms. I don't remember very well, but some people try to look to the limit that you can cut them. And I think the number of pieces that you can cut them, they are very tiny flatworms. And it was crazy. But it seems there is a minimum amount of tissue that you need to regenerate. And it also depends on the species, because flatworms is not only one species, there are several. And some flatworms are better regenerated than other ones. But even the ones that are better regenerate, I think it's a very tiny fragment, like few cells. But I'm not the expert, maybe I'm wrong. Yeah, so basically you cannot kill them just by cutting, in principle. You can try hard maybe if you want.
That's excellent. And yeah, next question is for Francesca. It says, were the algae inside the body of the coney, and how did this happen? How did this evolutionary process happen? And what is the advantage of the algae?
Yes, so while they are really inside, the skin of the animal, and actually I tried to get them out, and it's pretty hard without damaging the animal. And how it happens, we don't really know, but maybe it started. And what's the advantage? So for the animal, the advantage is easy, so it can get like oxygen and carbon and nutrients from the algae, from photosynthesis. And from the algae, I can only speculate, but it could be that they move, for example, so they can always have access to light. Or they need the CO2 that the animal produces. So yes, that's it.
We have another one for you, Francesca, from a person called Francesco. So many Francescas and Francescos in this network. So the question is, are you studying are you more focused on reproduction alone? Are you trying to make comparisons between sexual and asexual reproduction, or are other meanings behind studying these two different reproduction systems?
So I'm actually more focused on the sexual reproduction because I want to look at how cell types arise during development. So at different stages of development and then in the adult. But then, as I said, we don't know much about these animals. So when we get data from the cells, we can get a lot of more information. So it would also be interesting to compare sexual and asexual reproduction.
Thank you. Okay, we have more questions for Alba. Thank you everyone for the questions. So the question says, this regeneration pattern you observe in Pariale, the first shrinking and then reforming the last limb, is that a pattern that you can observe in other crustaceans, for example, crabs, and is this a common pattern in crustaceans?
Indeed, I don't actually know, but I could expect to also see it because crustaceans, as for example, also arthropods, when they regenerate, they do it within the exoskeleton. And at the end, you need to create, because the tissue is already there, you need to create some space. So I will expect to also observe the same.
Yeah, that would make sense. So we don't have any more questions presently, but yeah, we have a couple more minutes left, so please keep them coming.
There is a question from Enrico.
Is there?
Yeah, how do you decide what is a cell type and what is not? Are they specific to each animal or group of animals? I think directed to everyone.
I can maybe answer that. So yeah, I mean, there are different techniques to identify them. And recently, I mean, we can go very specific with like single cell RNA sequencing. So we can actually sequence all the active genes inside a cells. And that way we can see which specific genes are expressed there. And then, I mean, with the technique that I show, we can directly see where those expressed genes are located inside the animal. So we can also see at the morphological structure. We have also like electron microscopy data. And I mean, yeah, there are some cells that are specific to some animals groups, but some others have a lot of things in common, like muscles. They have a lot of yeah, the structure of the muscles, the proteins that they use for contractions, or for example, also photoreceptors to perceive the light and transform that energy into an electrical signal. I mean, the processes are kind of the same. The molecules might change a little bit, but we are able to compare them. Yeah.
It does depend, though. There isn't, I don't think there is a universal definition.
I have a question for Petra. And Petra, you mentioned that some animals can and some cannot regenerate the germline. Can you tell us a bit more? Do you know what determines that?
Right, so it's usually the animals that are able to regenerate missing parts of their bodies that can also regenerate the germline, because there's a balance between keeping the animal alive and making sure that there's enough energy to produce the progeny, so to invest to the other generation. And what determines that is basically whether there are some stem cells or not, and how much the animal makes sure that there is a separation between body cells and germ cells. Because in these exceptions that I'm not sure I mentioned, the germ cells arise quite late in development, and that's when the body cells also get their say in where they get specified. So it depends. The short answer is it depends on development process.
Thank you. I have another question for you, Petra. Then, so in your talk, you mentioned how many animals will set aside their germ cells and make sure that they don't mix up with the body cells. And is that the case in your animal as well?
This is something I'm actively trying to figure out by looking at gene expression that Francesca explained. And I'm looking at the genes that are also expressed in stem cells to figure out whether my animals have stem cells that could give rise to germ cells in their adult. So that's how I try to approach whether there is this separation or not.
It's interesting. We'll stay tuned and find out. We have another regeneration question that is a popular topic. So how could it be possible, and what levels would humans be able to regenerate? In brackets, maybe not a leg, of course. One day.
Yeah, or not. But okay, humans can regenerate to some extent. It's not true that they don't at all. We can regenerate the liver, for example. So it's not as spectacular of legs. And I mean, I don't know whether we will be ever able. I think this is very far away, but I'm sure, and this is not my field because I'm more dedicated to evolutionary questions, but I'm sure that people working in regenerative medicine, what they are attempting to do is to apply this to humans. And in the sense that I'm sure that maybe one day we will be able not to regenerate a leg, but to regenerate internal organs, as we do with the livers, and I think that will be very useful, for example, for transplants, because nothing else will be more compatible than your own cells that are regenerated another organ from another person. That will be very useful. But I don't know how it's going, honestly, if we are that far away. I hope not.
I hope so as well. YouTube question for Petra. And it says, are there many similarities between human germ cells and germ cells of other animals, even very distant similarities?
Oh yeah, there are very distant and well-conserved similarities in germ cells across pretty much all animal kingdom, and that allows us to look at them more closely. So when we choose these genes that are conserved in older animals, we can find germ cells in animals where we don't know where they are so far. So yes, there are conserved things, but there are also differences. And specifically when it comes to human germ cells, they express as well a set of genes that are well conserved. Yes.
Yeah, good to know. Yes, so surprisingly, we have more regeneration questions. And what about mistake multiplication and cancer with regeneration? So is there a relationship between regeneration and basically cancer growth?
I was mute, sorry. So I think So regeneration at the end is regrowing a body part, and cancer can occur when during growing there's no control, and so basically the body cannot control that. I think it has seen, but maybe I don't remember properly, that some animals that can regenerate are more resistant to cancer, and that could be because they control very well how to regrow body parts. I don't know if that answered the question.
I think it will do. I have another one for Petra. It's when you look at germ cells, it makes the most sense to look at adults, right? The stages that use the germ cells, but you need the offspring as well in your research. So I'm wondering what you use them for.
Well, yeah, like I said, because we don't know whether in my research animals where the germ cells come from, I need to look at earlier developmental stages, so early on in their life cycle, to see whether there is already something in there that could be a germ cell or not. So what I will do now with these 2-month-old guides is do some stainings, just like the ones, well, pretty much similar to ones that Luca showed you on the microscope.
Thank you. Okay, so that I think is a good note to leave it on, and we're a little bit over time now as well. So yeah, to wrap it up, thank you everyone who joined the talk, and everyone who asked question in particular, and in particular Sarah, who asked the question about cutting up the flatworm in infinite pieces. I really enjoyed that. And thank you to the speakers for their great talks and answering all the questions. And Sarah, do you want to say something for the end as well?
No, just also very big thank you to everyone who made it here tonight, and hopefully we'll see you tomorrow evening as well. Tomorrow at 7 o'clock we have another talk, and it's called The Secret Life of Sea Creatures, which I'm also very much looking forward to. Okay, well, with that, thank you.